Today we're going to discuss nucleic acids. So these are probably things that you've heard of before, at least I certainly hope so. We start with something called DNA, and that stands for deoxyribonucleic acid. And of course, that's the genetics code of cells, and that's found in the nucleus of the cell. You've probably also heard of RNA, which is ribonucleic acid. So this uses genetic code to basically synthesize proteins, but really it does a lot of other stuff too. There's different types of RNA. So DNA and RNA are part of a subclass of compounds called nucleotides, okay? So nucleotides consist of three things. We have a nitrogen-containing base, a 5-carbon sugar. Hey, you remember all those carbohydrates? You needed to know that for this stuff, okay? And something that's just a phosphate group. Now, we've talked about phosphate groups in the past, but this is kind of what they look like. You've got a phosphorus in the center. We have an oxygen on all 4 sides, and 2 of those oxygens are negatively charged. And then we have that connected to something. So there's different kinds of these nitrogen bases, and there's different kinds of these sugars. So let's talk about that. The nitrogen-containing bases, there's 2 major types. We have what's known as purines, and we have what's known as pyrimidines. So purines have a 2-ring structure like what you see right here. Now, I didn't draw out all of these specifics of what they specifically look like, but those 2 types are adenine, which is an A, is what we abbreviate a lot, and guanine, or we abbreviate that as a G most frequently. You also have something called a pyrimidine. So pyrimidines have a 1-ring basic structure that looks like this. And there's 3 types of these guys. So we have cytosine, which we abbreviate as a C, thymine, which we abbreviate as a T, and uracil, which we abbreviate as a U. So it's important to note, DNA uses A, G, T, and C. Uracil is not present in DNA. But RNA substitutes thymine with uracil. So you actually have A, G, U, and C in RNA. Why? Because that's what happens. It's just what happens, okay? So let me move this up a little bit. And let's talk about these sugars. So this is what differentiates DNA and RNA. Now, just so you know, these 2 structures here, I literally copied it over from one to the next. These are the same except for one spot, the spot that I have highlighted. So deoxyribose has an H right there on the 2nd carbon versus ribose has an OH. So they call it deoxy because there's no oxygen, D meaning not. And then the ribose is right here. So these both are sugars like you've seen in the past. Notice that I have highlighted not highlighted, I have marked the numbers for the carbons here as 1', 2', 3', 4', and 5'. That's going to be very important. 1', 2', 3', 4', 5'. Just like we did before, the only difference is now we put this little like dash up above those numbers and we say prime. I don't know why, that's just what we call it. So that's what the major components then are of DNA and RNA. Now, I told you that nucleotides consist of 3 things. Well, there's this other subcategory, and that's called nucleosides, and they're easier to build, but because they don't contain phosphate. So I'm going to show you what the nucleoside looks like, and then we can add in the phosphate and add a nucleotide, okay? So a nucleoside has a base at the 1' position. So it doesn't have to be the specific one. It could be any of those bases, nitrogen-containing bases that we talked about there. So A, G, T, U, or C, any of those would be fine. And so this is what a nucleoside looks like. Again, I literally copied and pasted this over here. This is a nucleotide. In this case, we have the same thing here. We have a base at the 1' position, and at the 5' position, we have a phosphate. So I don't want you to get confused in the fact that sometimes the double bond of the oxygen goes up on phosphate. I'm just pointing this out. So we could draw this as P with a double bond going up. If you drew it instead as a P with a double bond going out and a negative charge on the top and the bottom with an OR, it doesn't matter. It's the same thing. Just making sure that we all see that. This is a double bond right here, so sorry about that if you can't see it very clearly. Okay, so the difference in a nucleoside and a nucleotide is literally just the phosphate group. That's it, okay? So now let's look at what happens when we add those phosphates, because this is incredibly important when it comes to energy, and we're going to look at that pretty soon. So here I'm showing you at first what's known as Deoxy Cytosine Monophosphate. Okay, well, let's talk about what that means. Deoxy means that you're starting with DNA type of stuff because there's no OH right there. Cytosine tells you that you've got a cytosine up here. That's what the base is. And then monophosphate means, mono meaning one, says that there's one phosphate group over here. Okay, so we abbreviate that DCMP for deoxy cytosine monophosphate. The next thing that I'm showing you, though, is that we can go and add another phosphate group right here. And really it only adds this much because, well, this oxygen right here is combined with some hydrogen and that gives you water. So there's only like one little oxygen that's connecting those phosphates right there. But that's the only difference in what the structure is in the left versus what I just showed you right here. So can you see how we have two phosphates that are aligned here together or added together? Well, that's what we call a diphosphate. So this would be deoxycytosine diphosphate because we still have the deoxy and we have cytosine up here, right? So this would be abbreviated DCDP for deoxycytosine diphosphate. Okay, now finally, as you might imagine, we can add another phosphate group. And so now we have this whole line of phosphates with double bonds going up and these coming down. Now it doesn't matter again where you put your double bond as long as they're there. This would be deoxycytosine triphosphate. We still have a deoxy here. We still have our cytosine. The only difference is we've got 3 phosphates in a row. And this is DCTP is what we would abbreviate that as. So that's what it looks like when we're talking about a triphosphate, diphosphate, and monophosphate. The only difference is the number of phosphates that are connected. That's really important when we move forward. So I want to make sure that everybody understands those things, okay? Okay, so nucleic acids and like DNA and RNA are linked together by these long strands. That's really what kind of makes polymers like plastics too. Okay, so we talked about what the numbering system was, the 1', 2', 3'. Well, here's why we need to know that in part. The 3' OH of one sugar connects to the phosphate on the 5' of the next nucleotide. And that's what we define as a phosphodiester linkage. Well, let me show you what that looks like, okay? So if I scroll out here, there we go. Let's see if I can get all that on one page. There we go. So here's what happens. Let's look at the top part first. Let's look at this guy first, okay? So as I said, we're looking at the 3' OH of one sugar connects to the phosphate on the 5' of the next nucleotide. So if I have this guy connected to this guy, and I have some acid floating around, what's happening here is that the OH right here is connecting to the phosphate up here, right? And so one of those oxygens stays, and that's where this oxygen comes from right here. And so if you want to think of it this way, the OH here and this hydrogen make water. So this oxygen would stay, and that's what your linkage is going to be. Notice that this is coming off of the 3' down below. The phosphate is coming off of the 5'. So that's what we say, the 3' OH of one sugar connects to the 5' phosphate of the next nucleotide. That's where these come from, these linkages right here, okay? You might need to go back and look at that a few more times to really understand what I just said. That is okay. Make sure that you know how to put those together, because this is really important. When you link these together in a chain, I've highlighted for you what things look like. Like at these highlights, that's your phosphodiester linkages. And so you literally get like this chain that's sort of going down. And so we refer to this as the 5' end because at the 5' carbon right up here, it's empty. There's not another linkage there, right? And this is the 3' end right here because, again, the 3' end is kind of empty. So we call that the 3' end. And this is an example of what DNA structure would look like. Of course, you're going to have different bases that are here, and that's okay. The other thing is that when it comes to DNA, the bases are linked to something else, and then you have another strand that's going up the opposite direction, which we'll talk about next time. Okay, thank you all, and I'll see you on next video.